Welcome to part 2 of chapter 12, talking about gene regulation in prokaryotes. In part 1 we gave you a story of the lac operon and its mutations. In part 2 we're going to talk about the tryptophan operon and also how the regulation of a bacteriophage takes place. And in between we have a sprinkling of other types of control, including riboswitches. The logic that we described for the first story, the lac operon, and the introductory parts of the previous video are going to be resonating again as we move through these two stories in this video. Your job is to make sure that you understand and predict the logical outcomes as we present information. In particular, contrast the two systems, the lac operon and the tryptophan operon first. Notice what's different. There are major differences that dictate the outcomes. Understanding those differences is key to remembering which system should be applied with which knowledge. One does not want to mix up Little Red Riding Hood story with Jack and the Beanstalk. Let's tell you the first major difference between the second story and the first. In the first story, we were looking at the regulation of the lac operon, the lactose operon. So lactose is a food molecule which is broken down by catabolic processes inside the bacterial cell to release energy, which is then used to power the cell, and that energy is in the form of ATP. In addition, the lactose operon was inducible, and it used both positive and negative control mechanisms to regulate its outcomes. The tryptophan operon is almost the reverse. Tryptophan is an amino acid that's made through a biochemical pathway by starting with simple molecules and reconstructing them using a set of enzymes. So in this case, this is an anabolic pathway. So there's a great difference right there. The first one broke down something. This one is building up chemistry within the bacteria. Tryptophan is the amino acid that's needed to construct polypeptides by ribosomes. So this is an essential component. If something is essential, then the chances are that pathway is normally on. But if the cell has enough tryptophan from other sources, i.e. importing it from your digestive system, then the bacterial cell can turn this system down or turn it off to save energy. And the last thing is, the tryptophan operon can be attenuated, and that means it can be finely controlled to suit the circumstances within the cell. Attenuation applies to the dimmer switch that we spoke about earlier. So if it's running at 70%, you can reduce it down to 40% or 30% in regard to the activity of this particular set of genes. When something is turned down or turned off when not needed, use the term repressible. So we shouldn't be surprised to find two ways in which this operon can be downregulated or turned off. Before we look at the way this regulation takes place, let's look at the general architecture of this part of the bacterial chromosome. The tryptophan operon, like all other operons, consists of more than one structural gene under the control of one switch. In the case of tryptophan, the 5 structural genes are attached to a regulatory region, which has a promoter, an operator, something new called a leader sequence that contains the attenuator region. So we'll look at this in a second. And the names used are similar. We have the trpP, that stands for the promoter region, the trpO, which stands for the operator region, and the trpL, which stands for the leader region. The 5 structural genes are labeled trp something, and they're actually labeled backwards, E, D, C, B, and A, with regard to their positioning along the operon. And they're responsible for producing the enzymes necessary for taking the raw material and converting it to the amino acid tryptophan. There's a 6th gene which resides outside the operon, and that's called the trpR, for repressor. And that protein, product of the gene, is important in binding to tryptophan to do something, as we'll learn shortly. Figure 12.13 presents a nice overview of the tryptophan operon. Let's start with the structural genes. The trpE2A are located right here. Just upstream of them we have something called the Attenuator Region that we didn't see with the lac Operon. Then we have other parts which all belong to the leader region, and we're going to look at that in detail a bit later. Then we have the operator region and then the promoter region. The result of transcription is that transcription starts at the +1 site, and then if nothing changes that behavior, it'll continue all the way through until the end of the last gene, trpA. And that polycistronic messenger RNA will then be attached to by ribosomes at different points. The translation process will produce these polypeptides, and the polypeptides will then convert themselves to enzymes, and those enzymes will drive the production of tryptophan, starting with coricimate. Looking at this biochemical pathway, it makes sense to make these enzymes using a common switch because they're all needed in equal proportions and at the same time. We didn't exaggerate on this region here in the leader, but rest assured we're going to talk about this immediately. Let's begin by talking about the trpR gene, which, as we mentioned earlier, is not part of this operon. It's located elsewhere on the chromosome. The product of the trpR gene is called a repressor, and that repressor will bind to two things. It will bind to tryptophan at one place, and it will also bind to the DNA at the operator at a second place. So it has two binding sites. So because tryptophan binds to the repressor and it has an effect on the rate of activity of this operon, we consider tryptophan, the food molecule itself, to be a co-repressor of the tryptophan repressor. And in order to activate the repressor to do its job, which is to prevent transcription, the tryptophan has to bind first to the repressor. So without tryptophan, the repressor is just floating around the cell aimlessly without anywhere to go. But when tryptophan binds, it then is targeted directly towards the operator region of the trp-operon. Just move up and take a more realistic and general view of what's happening. Use logic. What we're saying is that when tryptophan is present inside the bacterial cell and it's produced by this pathway, there must be enough tryptophan to spare to bind to this repressor. So tryptophan comes back from the end of the pathway where it's made, and it interacts with the proteins that are going to turn on this operon. This is a great example of a negative feedback loop. The tryptophan controls its own destiny. When tryptophan levels are low, there's not enough to bind to the repressor, and therefore the operon should be turned on. When there's too much tryptophan in the cell, to save energy, the operon will be turned off. So never forget the overall perspective of why you're learning something. Let's now look at in detail what's happening with respect to the switch that controls the activity of this system here. So the trp-operon is transcriptionally regulated by the repressor. The repressor protein represents a two-way switch. Either it can or it cannot bind to the operator and block transcription. In its naked form, it cannot bind to the operator. Therefore, it needs to have a partner, and the partner happens to be the tryptophan co-repressor. So the binding of the tryptophan causes a conformational change at the DNA binding domain of the repressor, and is able to bind operator at that point only. Should the tryptophan come off because of low levels in the cell, then the repressor protein will be removed off of the DNA at this operon. And if we dive back to this slide 12.2, we'll see that it works in this manner, which is a positive mechanism of control over transcription. So the effector molecule will be the tryptophan, and the protein, the activator protein, it will be the tryptophan repressor. If you look carefully, you'll see the second major difference between this operon and the lac operon. In the case of tryptophan, it has to be absent in order to prevent the activity of this operon. In the case of lactose, it has to be absent in order to prevent the repression, the blockage of the operon. So it works in the opposite way. When the early scientists performed experiments with the tryptophan operon, they were surprised to find that it didn't behave as logic dictated. Mutational analysis revealed a strange outcome. In cells where the tryptophan R gene, the trpR, the repressor gene, was defective, one would expect that if you don't produce the repressor, then there's no way it can bind to the tryptophan because it doesn't exist, and therefore you should never get this type of repression. And if you can't block by binding to the operator, there should be no way of preventing transcription. And in those cases, they were expecting to find that tryptophan was expressed constitutively all the time. But they didn't see this. What they discovered in these trpR- strains, they had a higher rate of Operon transcription in the presence of tryptophan than trpR plus strains. Table 12.6 shows this data. So if tryptophan is present inside the cell, then we do not need to make any more tryptophan. So the wild type cell, the one that has a normal trpR repressor gene, is active at this leaky level of 8%. But for the same cells, when tryptophan is absent, then they need to start making their own tryptophan, and that switch turns up the operon to about 100% of its capacity. In the trpR- mutants, the same thing happens when tryptophan is absent. The system turns up to 100%, but when tryptophan is present inside the cell, it's no longer leaky at 8%, it has a significant activity. So the difference between the two suggested that something else was also controlling the tryptophan operon. This second mechanism turned out to be a marvel of biological engineering. It really is a fascinating system that we should all learn, because it just shows you the beauty at which the nucleic acids have evolved to interact with the environment. I cannot but still be flabbergasted by how this system works. To fully appreciate and also understand how this second form of regulation works, we have to focus our attention on the tiny part of the chromosome that we didn't really pay attention to previously. So if we go back here, we're now going to focus on this region here in the leader sequence. I've borrowed this slide from another source just to indicate where we are. So we have the start of transcription, the +1 back here, and then this is the very first part of the RNA that's produced from the gene, and you can see that it has +50, +70, +110, and +130, 140, and then 160, and then you get into the first gene here, the AUG of the trpE polypeptide. The region just upstream of that is called the Attenuator region, and this is where we're talking about. So this region here is going to be very important in controlling the method by which this operon is regulated. Looking at the linear arrangement of this regulatory region, we now find that we know where the promoter sits, where the operator sits, downstream of that. Then downstream of that we have the leader sequence, and then we have this very important region within the leader sequence known as the attenuator sequence. And then following the attenuator sequence, we have the structural genes. So we'll be looking at what happens in this region here during the passage of RNA polymerase along the DNA. In order to fully appreciate how attenuation works, and then to understand this statement here, we simply have to go back and look at this table. And the table says that in the presence of tryptophan, the Operon is leaky, still operating at 8%. So understanding that is very important. Then this statement becomes apparently clear that even when tryptophan is present inside the cell, and logic tells you you need to shut down this Operon because you have enough tryptophan, the leakiness allows the production of certain products from this Operon. The transcription of the leader region always gives rise to the production of this RNA molecule, messenger RNA. And the messenger RNA has along its length some very important elements that are responsible for deciding what to do. And either it will terminate transcription, or it will allow transcription to continue into the structural genes that are located downstream. So this is another switch. And the way the switch works is marvelous. So let's look at the structure of the messenger RNA. So along the messenger RNA, we have a AUG codon, and then further downstream we have 2 codons for tryptophan. Very important. Then we have 4 regions that are repetitive, and they can form structures amongst themselves. And then we finally have a poly-U region before we get to the end of the leader sequence. So this arrangement is very important. The actual length of the leader region of the gene is 162 base pairs. And within that region, there are 4 repeat DNA sequences. We just spoke about them. And the messenger RNA produced contains complementary repeats, as well as start and stop codons for a 14 amino acid polypeptide. The 4 repeats on the messenger RNA, remember, we're not talking about DNA at this point, we're talking about messenger RNA. The 4 repeats on the messenger RNA can form different stem loop hairpin-like structures with each other. And that's the key. And we're going to look at that in a few seconds. But as we mentioned earlier, just upstream of them, we have the 2 back-to-back codons for tryptophan. And these are the sensors that drive the decision of what to do. And they depend on their functionality for the availability of tryptophan. So if tryptophan is available, the sensors will do one thing. If tryptophan is absent inside the cell, they'll do a second outcome. Thus, the leader sequence, if made fully, can fold, the RNA can fold into this double stem loop structure, where we have the beginning tryptophan codons here, then region 1 and 2 snap back using complementary base pairing to each other. Likewise, region 3 and 4 can do the same. And then the poly-U's are here. Under different circumstances inside the cell, the actual arrangement of this structure is not like this, but like this. So in one scenario, we get a 2-3 stem loop structure, and in a different scenario, we get a 3-4 stem loop structure. The decision as to which of these 2 will be formed depends on the presence or absence of tryptophan. Because we're dealing with prokaryotic cells, there is no nucleus. And as you remember, or you should do, the transcription and translation processes can happen one after the other. So as soon as a small amount of the messenger RNA is made, a ribosome is able to bind at the same time as transcription continues to this messenger RNA. And the question is, where does the ribosome stall or not stall during its translation process? So if the ribosome stalls at one point, one structure will form. If the ribosome stalls at a different point on the messenger RNA, a different structure will form between the polymerase, which is over here, and the ribosome. So there's an interplay between these two protein machines. This figure is designed to help determine which of two outcomes is going to be feasible. Let's start with the top one. When there's high levels of tryptophan inside the cell, then that tryptophan is available for the ribosome to encode a polypeptide. And as the ribosome travels through the messenger RNA region calling for the tryptophan to be incorporated, remember the two tryptophan sites that we spoke about earlier? These two. So here the ribosome will have no problem passing through, because when it calls for tRNA with tryptophan, it's delivered because there's enough tryptophan inside the cell. So the ribosome will not stall in region 1. It'll proceed quite rapidly and cover region 1 and begin to impinge on region 2 when the RNA polymerase makes region 4. So RNA polymerase is making this part of the molecule right now. And because 2 is unable to bind to 3, 2 is covered by the ribosome, that region is not available. So 3, even though it wants to bind with 2 to form a hairpin, it can't, because the ribosome is covering that region. So it finds the next best alternative, which is it waits for 4 to be produced, and then it binds to 4. And that forms the 3,4 hairpin. And that 3,4 hairpin is sufficient in tagging off physically the RNA polymerase off of the gene Operon of the tryptophan. So this snapping together of the 4,3 region is a physical force which pulls the RNA polymerase off of the DNA. We've seen something like this in another form of attenuation somewhere else, but here it is in actual use. And that result means that there's no need for the RNA polymerase to go further into the structural genes, and therefore the Operon is terminated early. Basically, there's enough tryptophan, why make more? The converse of that is illustrated here. And again, it's a very simple logic. As the RNA polymerase makes these regions, and it's right here, the ribosome follows. But because there's not enough tryptophan inside the cell, there won't be enough available for the production of tRNA molecules, transfer RNA molecules that are loaded with tryptophan. So there's a deficiency of tRNAs carrying tryptophan. Therefore, the ribosome is forced to stall, wait, until it gets a delivery of tRNA carrying tryptophan. And that's not going to happen. So the ribosome just sits here, covering region 1. If region 1 is covered, and then region 3 is made, region 3 will snap onto region 2 at a higher preference than it would bind to region 4. Because of the soft nature of this loop, and the complementary base pairing formed between 2 and 3, as well as the distance of this loop from the RNA polymerase, the force of tugging on the RNA polymerase is not significant. And therefore the RNA polymerase is able to continue its journey into the structural genes all the way to the end of the Operon. So everything hinges on the delivery of tRNA carrying tryptophan. And if there's not enough available, you get one outcome, this one. And if it's available, you get the second outcome. And that makes logical sense. This turns off the Operon, and this mechanism here turns on and keeps on the Operon. This form of attenuation is very sensitive to the tryptophan levels within the cell. Since I have these slides available, might as well look at them. The top half provides us with the outcome of the 23 loop too far away to affect transcription. Transcription will continue. And the reason it does that is because the ribosome will stall on the messenger RNA. In the bottom half, we have termination of transcription by a mechanical pulling effect. And that's because a 3-4 loop forms due to the fact that the ribosome has covered up the region of 2 that would normally form with 3. This is the equivalent figure from our textbook, and it shows the potential of the messenger RNA leader sequence to form secondary structure. And down here we can see the alternative forms of secondary structure that are made depending on the positioning of the ribosome and the RNA polymerase. So in summary, the formation of different loops is very important and key to the effect on the RNA polymerase. And as we just said many times, the 3-4 stem loop is what causes termination of transcription and halts the RNA polymerase from going further. The function of the polyuracil region along the messenger RNA is designed to slow down the RNA polymerase a little bit, so giving the option to the ribosome to do its job. Figure 12.16b gives us a pictorial representation of a ribosome encoding some polypeptide which is following along on the leader sequence. If one looks carefully, you'll see there's a stop codon downstream of the 2 tryptophan codons. Once the decision about attenuation has been made, we don't want this ribosome to keep following along the messenger RNA into these genes. We want it to stop, and that's exactly what happens. Once the system has performed its job, the RNA is translated into a polypeptide that's then degraded, and the stop codon prevents the ribosome from proceeding any further, and the system then self-regulates. Mutational analysis has helped us understand how this system performs. One way that you can understand this is to mutate the tryptophan codons. If you mutate one of the 2 codons, that does alter the attenuation responsiveness to tryptophan. But mutating both of them will abolish the ability to attenuate, and then the ability to sense tryptophan is lost, and the ribosome will never stall, and it will continue on and form the 3-4 loop that will pull off the RNA polymerase. Any mutations in the 3 or 4 region which prevent a stable hairpin structure from forming between the messenger RNA, that will reduce or eliminate the efficiency of attenuation, because you're reducing the mechanical force that the snapback of the hairpin causes on the RNA polymerase. Figure 12.17 shows us some of the experimental changes that have been made along the 3-4 region. This mechanism does not appear to be unique to tryptophan. Indeed, many other biochemicals that are synthesized through anabolic processes within the cell seem to have utilized the same system. So if we look at other operons, phenylalanine and the histidine, we also find that they contain in their leader sequence a requirement for the same amino acids as is being regulated by that operon. And mutation of these sites will lead to a dysfunction of the system. Other examples of bacterial operons for different amino acids are also presented in figure 12.18. Bacteria have evolved to respond to their environments using various mechanisms depending on the urgency of the response necessary. Sometimes the transcriptional level of control may not be quick enough to influence the behavior of a protein that's necessary in the cell. In those cases, it's quicker to have translational control. so that you can control the production of a protein or not at that level. The further one is away from the end, the longer it takes to achieve the end point, which in most cases is a protein. But don't forget that in a large number of instances, we don't need to make proteins. We're dealing with transcription and making some type of RNA molecule. And that RNA molecule can have a direct impact on the survivability of that cell. We should recall that bacteria only have one type of RNA polymerase for transcribing all their genes. However, it has a partner known as a sigma factor, and the identity of the sigma factor depends on from which gene that sigma factor is made. So there are multiple genes for making multiple sigma factors. And the idea behind this is when a cell becomes stressed, it will express different sigma factors, therefore changing the soup of genes that are activated at any particular time. So a great example is heat stress. So heat stress is normally when the bacteria find themselves at a higher temperature than what they normally expect. So our guts are at 37 degrees, and some bacteria can be placed in test tubes at 45 degrees. And we observe that those E. coli cells generate alternative sigma factors, and they activate expression of specialized heat stress genes which protect the bacteria at the higher temperature. And these heat shock genes, as they're known, encode proteins that protect cells from certain types of heat-induced damage. At these higher temperatures, the normal sigma factor, rho 70, is ineffective. It is unstable, and therefore an alternative factor known as rho 32 is used. And that combines with the RNA polymerase to turn on some genes that are similar, other genes that are different. One major difference between sigma 70 and sigma 32 is the recognition point on the DNA. Sigma 32 recognizes promoters that contain these GC-rich regions at the -10 position. Now, GC is held together by 3 hydrogen bonds rather than the usual AT, which is held together by 2 hydrogen bonds. So at the higher temperature, the GC-rich regions are more stable than the AT-rich regions. From where does the sigma 32 come from? It's a protein, so proteins should have their own genes. Thus, sigma 32 is made by a gene called RPOH. The promoter for RPOH is recognized by sigma 70, containing RNA polymerase, when the temperature is elevated. Indeed, other sigma factors are also produced inside the cell when the temperature begins to change, and a third sigma factor, called sigma 24, is also elevated, and that binds to RNA polymerase, which contains this factor, also increasing the transcription of RPOH. Thus, the stimulus for changing the biochemistry of the bacteria is the temperature in which the cell finds itself. So there's your logic. If you drop the temperature, the system will revert back to the original use of sigma 70. Figure 1219 details what we just spoke about. There appears to be an extra layer on top of this regulatory system that makes logical sense also. So scientists have discovered that there are some chaperone proteins that are present inside the cell, and their activity is controlled by the temperature also. So at normal temperature, these chaperones bind to any small amounts of sigma 32, the heat shock sigma, and prevent it from binding to the RNA polymerase. So they block its active site for RNA polymerase. As the temperature rises, these chaperones are naturally released because of molecular vibrations from the sigma 32. Then sigma 32 is free to join RNA polymerase core enzyme and form the holoenzyme, and therefore go and find its target sequences. But the chaperones aren't wasted. They're redirected to limit heat damage by binding with other proteins. When the temperature drops, they release from these other proteins, and they come back and reassociate with sigma 32. A wonderful system. As mentioned at the beginning of the chapter, in bacteria, the preference is for transcriptional regulation. But there are cases of translational regulation. And there are two mechanisms that have been discovered so far. In one binds protein to the messenger RNA to prevent its translation. So the protein blocks messenger RNA from being combined with a ribosome. In the alternative situation, it uses complementary antisense RNA to bind and block messenger RNA translation. So the formation of double-stranded RNA is enough to block translation. Under the first type of control, proteins bind to the messenger RNA close to the Shine-Dalgarno sequence. Remember, the Shine-Dalgarno sequence is where the ribosome will bind to the messenger RNA and then seek to find the AUG start site. So if these proteins are binding close to there, they will interfere with the interaction between the messenger RNA and the 16S RNA small subunit, because they come together first. And if that does not assemble, then the whole ribosome will be prevented from assembling and doing its job. In a similar way, the ribosomal proteins in E. coli are regulated in the same way from each operon. So one of the products of the operon, one of the gene products, the protein, will bind to the ribosome and prevent the ribosome from translating the messenger RNA. So what we're witnessing is that there are multiple levels and multiple ways that evolution has equipped biological cells in achieving outcomes. The second mechanism uses something known as antisense RNA. And that's an RNA molecule that is complementary to some part of the messenger RNA. And the binding between the two, the messenger RNA and the antisense RNA, creates a double-stranded region of the messenger RNA. RNA itself can act as a switch. Under one set of circumstances, it will form one secondary structure. In an alternative set of circumstances, it will form an alternative structure. One is conducive to regulating the behavior of transcription, the other may not be. So this system has been expanded upon since its discovery in the early 2000s. These riboswitches tend to be more prevalent than we ever thought. In some systems and situations, they regulate transcription. In other situations, they regulate translation. And they can also regulate messenger RNA stability. The next three examples illustrate how riboswitches function. There's a commonality to the method, and we'll talk about that at the end. So the first example is to do with Bacillus subtilis, a bacteria that has many operons, one of which regulates the production of thiamine pyrophosphate, TPP. One could predict that the end product, the TTP, may have something to do with regulating the operon itself. And indeed, in this case, it does. And the way it does it is to bind to a riboswitch regulatory sequence on the messenger RNA at the onset of the thiamine operon. In circumstances when the amount of TPP is low within the cell, there's not enough available to bind to the riboswitch, the messenger RNA. And the riboswitch forms a structure which encourages transcription to continue. However, in the alternative, when TPP is available in significant concentration, it interacts directly with the structure of the riboswitch, forming an alternative termination structure. And this hairpin loop here is known as the termination stem loop, and that has an effect on the RNA polymerase by also doing what we learned earlier in tryptophan attenuation. It tags on the RNA polymerase, causing it to fall off, and therefore limits the production of more TPP. An example from E. coli allows us to see a slightly more evolved evolution of a riboswitch. In this case, the system is the same. When TPP is low in E. coli, the switch is inactive. and the binding site for ribosomes is made available, and they are able to bind and then translate. But in the case of high levels of TPP, the Shine-Delgado sequence is blocked by a hairpin loop, which prevents the ribosome from binding. Therefore, there's no translation. The difference between the two is that in the first example, we're interfering with RNA polymerase, transcription. In this example, we're interfering with translation with the ribosomes. In the third example, we have a slightly alternative mechanism of action. In this case, the Riboswitch is not functional at low levels of this enzyme system that produces sugar. But as the level of the final product increases, it comes back and then it can interfere with the Riboswitch. So this is another feedback loop, negative feedback. By binding to the Riboswitch, the presence of the protein causes cleavage of the messenger RNA. Therefore, the messenger RNA cannot be used for translation. The last story is the most complex to understand. And it's been decided that we really want you to have a general understanding of this, but not a deep understanding. So where do we begin? Well, the story talks about how a bacteriophage, a virus that infects bacteria, can opt to have two forms of life cycle. It can opt to either undergo a lytic life cycle, where it immediately produces progeny and destroys the bacterial cell as fast as possible to then infect neighboring cells. And this is good when food is plentiful, lots of bacterial cells, and the conditions are perfect. But should conditions become unfavorable, then the virus is able to adjust its life cycle so that it inserts a copy of itself into the bacterial chromosome for a future lytic cycle. So it can be transmitted from one cell to its progeny through binary fusion of bacteria, and then many generations later, when conditions become suitable, the phage will wake and kill and destroy and lyse the cell and continue its life cycle. And if it chooses that latent method, then it's called lysogeny or lysogenic cycle. Viruses which can choose as this are called temperate. And Lambda phage is the prototypical phage that geneticists used to examine how the system works. The essence is this. Phage Lambda is in the wild type condition, the normal condition, about 48,000 base pairs in double-stranded DNA. And it contains about 60 genes. Once that DNA is injected into a cell, it circularizes by joining its ends together because they have 12 base pair overhangs. Those are called cohesive ends because they're sticky in terms of chemistry. And the genome is organized into operons. And those operons are expressed either early, middle, or late during these infection cycles. Figure 1224 shows the arrangement of the genes along the circularized chromosome of phage Lambda. So the genome is organized into operons that function at defined times during the infection of the host. The cohesive ends, which I believe are here, will come together to form a circle of DNA once it enters the bacteria. And then there's a race to transcribe different genes. And that race is ultimately won by the prevailing conditions within the bacterial cell. If the bacteria is healthy, then one pathway will be followed, it wins. And if the bacteria are stressed, an alternative pathway will be followed, and that will then lead to lysogeny. So the activation of these early genes is broken up into two categories: immediate early and delayed early. And then we have the late genes, and they're expressed in the order depending on the conditions. Students are encouraged to view videos about the lytic and lysogenic life cycle of Phage Lambda online. For those who wish to know in detail the life cycle of this particular bacteriophage, then another video will be amended to the website. But for now, thank you for watching.